Okay, hello everyone. Welcome to today's webinar, which is titled Bilayer-Mediated Regulation of Membrane Protein Function by Small Molecules. For those who don't know me, my name is Tom Constable and I am the host for the webinar today. I joined Applied Photophysics in 2015 and have actively been a specialist for our Stop-Flow products in that time. Currently, I'm the product manager for the SX20 Stop-Flow and I'm also the customer support manager within the organisation. I hold a PhD in chemistry and I'm also a chartered chemist with the Royal Society of Chemistry, and one of the best elements of my job is being able to talk with and listen to and learn about our customers and their research. So today it's my great pleasure to introduce Dr. Olaf Anderson, who has kindly agreed to present his research to you all. Professor Anderson is from the Department of Physiology and Biophysics at Weill Cornell Medical College in New York. He's the director of the Weill Cornell Rockefeller Sloan Kettering Tri-Institutional MD PhD program. Olaf studied medicine at the universities of Aarhus and Copenhagen in his native Denmark. He then took postdoctoral positions at the University of Copenhagen and the Rockefeller University before joining the then Cornell University Medical College. His research interests include the energetic coupling between membrane proteins and their host lipid bilayer, bilayer-mediated effects of biologically active molecules, and the bilayer-dependent basis for cytotoxicity. Today he'll be talking about using single-mixing stop-flow spectrofluorimetry to quantify the bilayer modifying potency of bioactive molecules. I have a few maintenance points to cover just before we get started. The presentation will be around 40 minutes in length, with 10 minutes of questions to follow. If you'd like to ask a question, then please add them to the chat box on the side of the GoTo client, and I'll put as many as possible to Dr. Anderson at the end of the presentation. If we don't get round to answering your question, then we will respond to you directly in the next few days with the email address that you signed up with to answer your question. The webinar is being recorded and a link will be available in the next few days for you to view the webinar afterwards. At this time, I'd also like to invite you to attend our second webinar in this two-part series, which will be titled Mechanisms of Underlying Drug-Mediated Regulation of Membrane Proteins using Sequential Mixing Stop-Flow to Study Regulation of Membrane Protein Function. And this will be presented by Rada Rusanova, who is an Associate Professor of Research and Physiology Research in Physiology and Biophysics, also at Weill Cornell Medical College. That's scheduled for April the 8th at 11 AM Eastern Daylight Time or 4 PM British Summer Time. And if you'd like more details or to register, then please visit the Applied Photophysics website. So with all that said, and I'd like to thank Olaf for his time today, taking time out from his busy schedule to present the webinar. I will now hand over to Olaf to present the webinar.
Thank you very much, Tom, for the introduction, and good morning or good afternoon to all of you wherever you are. So what I'm going to talk about today is a question that has occupied me for many years, namely the bilayer-mediated regulation of membrane protein function by small molecules. And this is a project that has involved collaborations with many different people. And today I will highlight work done by the people whose names are in bold. Many others really contributed, but these are the people who contributed to experimental results to the slides that you will see. So the basic issue we are dealing with is the properties of the cell membrane. It is an elastic body. This is not a video, but there is, if you looked at the cell membrane, there will be undulations. You go in and out of the plane. There will be thickness fluctuations. Membrane proteins are embedded into the bilayer. Membrane proteins undergo conformational changes, as I will show you, and the energetics of these conformational changes are regulated in part by the mechanical properties of the host bilayer in which they are embedded. Sort of as a background, I think it is important to distinguish between two different kinds of regulation of membrane protein function by the lipids in the lipid bilayer. We can have lipid regulation, which involves chemically specific interactions between the protein and the individual lipid molecules. And I'm not going to discuss this at all. And then we have the lipid bilayer regulation, which is sort of the physical, non-specific interactions with the bilayer as a mesoscopic entity with collective physical properties. Up here, you can look at the regulation using concepts and language that you would use if you describe the binding of a ligand to a globular protein. Down here, we do not have that language to our disposal. It becomes really exclusively a question of thermodynamics, of changes in energy, energetic coupling between the bilayer and the host protein as the protein undergoes conformational changes. This is a question that has occupied people for many years, and there has been described using many different terms. The first that was used, and this was in the 1950s and 60s, was membrane stabilization. It was noted that some experimental maneuvers reduced the electrical excitability of nerve membranes or reduced the sensitivity of red blood cell membranes to osmotic license. Then the next term that came around in the early 1970s was bilayer fluidity. And over the next, say, 10, 15 years, so to say, the bilayer regulation of membrane protein function was described using a variety of terms that are somewhat difficult to quantify. And then came sort of in the early 80s, a new set of concepts appears, the notion of a hydrophobic mismatch between the hydrophobic lengths of a membrane protein and hydrophobic thickness of the host bilayer, which would lead to mismatches, would lead to bilayer compression, maybe expansion. There are other terms. And finally, in the most recent, we have membrane domain organization. All of these terms describe different aspects of the same problem, and Abraham Maslow long ago noted that I suppose it is tempting if the only tool you have is a hammer to treat everything as if it were a nail. And I think that applies to all of these terms, and equally it applies to what I'm going to talk about today. But if you take many studies on membrane proteins that you have purified, reconstituted into lipid bilayers of defined composition, then starting with work of Martin Caffrey and Jerry Feigenson around 1980 with the sarcoplasmic reticulum calcium ATPase, it became clear that systematic changes in the thickness of the bilayer would lead to changes in enzyme activity and transport activity. And many different proteins have been studied this way and show conclusively that changes in bilayer thickness affect changes in membrane protein function. Maneuvers that change the lipid curvature, where you change the tendency of bilayers from it being preferring planar organization to more curved organization, similarly change function of membrane proteins. But it turns out that whereas you can do fairly well-defined changes in bilayer thickness, if you change lipid curvature, it actually becomes impossible to avoid also changes in bilayer thickness. And so these results are a little more subject to interpretation. But the take-home message of all of this is that there really are two types of membrane proteins: those that are known to be altered by changes in bilayer properties and those that have yet to be studied. I here have paraphrased what was said about protein phosphorylation when I was a medical student in the 1960s. But here I show a specific example. This is a structure of an amino acid transporter, GLIT-PH, and it is studied by my colleague at Weill Cornell, Olga Budker. And she and her colleagues have crystallized this in different conformation where the protein is, the ligand binding site is facing the extracellular solution or facing the intracellular solution. And Leah Stanford, a student in my laboratory, together with a fellow graduate student, Naveed Paknejad, took Dr. Budka's structures and took these two structures, and then they color-coded them sort of as a plot where you see what is the extent of the conformational changes that occur between these two conformations. And there's a core of the protein that really doesn't move at all. And then you have three transport domains that are sort of hooked up to this central core that move substantially as the protein undergoes these conformational changes. And you can see the changes are large, and therefore there's going to be changes in the packing of lipids around the protein. There may be changes in the orientation of the protein, so to say, in the plane of the bilayer. And here I show another example, which is a mechanosensitive channel of small conductance from material membrane, which many years have known it exists in closed and open conformations. And you see that these conformational changes are associated with substantial changes in the bilayer spanning domain of the protein. Unfortunately, I don't show the bilayer with the same resolution as showed in the pH structure. And the important point here is that as they go between the closed structure, the lipids are going to organize different in this structure than in this structure here. In each conformation, there's going to be an energetic penalty associated with inserting the protein into the bilayer. There'll be the fairly rigid protein structure is going to constrain the movement of the acyl chains, which will give rise to entropic contributions to the energy, but there are other terms as well. And so we go between these two conformations with different bilayer deformation energies. And if you now look at say this as now more generally two different conformations of a protein, we would have a bilayer deformation energy associated with this conformation, and the free energy difference between these two conformations becomes a bilayer contribution to the overall free energy cost of the protein conformational change. And so, but it turns out that this is not all, because as also Dr. Rusanova is going to allude to in her webinar in a few weeks, if you look at this structure, you can see that the alpha helical segments are not perfectly aligned at the bilayer solution interface. And as a student in Harel Weinstein's laboratory, Jan Model, noted many years ago, that led to some polar residues in the protein being exposed to hydrophobic environment, and hydrophobic residues might be exposed to a polar environment. It is impossible to obtain perfect hydrophobic adaptation between the protein and the bilayer, and we call this term the residual exposure energy. This is, and I bring this out now because this turns out to be a likely pharmacological hotspot in the proteins, as I will also come back and discuss later. So what we are interested in is the energetics of structural changes in glit pH. Despite the advances in computational methods, we really don't have the wherewithal to study these now. And so what we have done for many years is to use the Gramocide channel, which sort of is the opposite extreme of complexity from glit pH. It is two subunits that form the channel. Each subunit has 15 amino acids. It is an alternating LD sequence, which means that then you wind this into a helix, then you actually have a lumen here that is large enough to allow monovalent cations to move from one aqueous solution to another. The channel is, as far as we know, ideally selective for monovalent cations, and there is a size cutoff. They have to be less than about 2 angstroms in diameter. But they can allow for very efficient movement of cations across a membrane. As you can see here we have the structure again. And because the channel is a dimer, it forms by transmembrane dimerization of 2 non-conducting subunits here. They will move around in each plane of the membrane by thermal diffusion. When they line up, a small fraction of these diffusional encounters is going to give rise to the bilayer spanning channel, and we can measure the formation of the channel by these small but well-defined changes in current through the bilayer. And it turns out that the hydrophobic length of the channel is on the order of 22, 23 angstroms. That is less than the hydrophobic thickness of the host bilayer. So channel formation leads to a local bilayer compression and bending of the 2 bilayer solution interfaces, which incurs a cost. And the bilayer responds to the formation of the channel by imposing a disjoining force on the dimer, trying to pull it apart. And that actually becomes the basis for what I'm going to tell you in the next few slides. Now, if you look at the simplicity of the structure, it turns out that with Gramocide channels, we do not have to worry about the residual exposure energy. So we really have that the Gramocide monomer dimer equilibrium will be composed of contributions from the rearrangement within the protein, which it basically becomes a hydrogen bond stabilized joint between the 2 subunits, and then the perturbation of lipid packing around the channel as it forms. And so what started me out on these experiments now almost 35 years ago is an experiment similar to the one I show here, where we, for reasons that I'm not going to discuss, decided to do a control experiment, basically looking for what were the effects of small amount of dirt on the function of Gramocide channels. And what better dirt than a detergent, at least in my laboratory. And so we added a detergent, Triton X100, to a bilayer, and we were expecting to see changes in the distribution of current steps, which we did see to some extent, but the dramatic effect was these are experiments obtained in the same membrane about 5 minutes apart. Here we see before we add the Triton X100 to the bilayer. Here we see what happened after we add Triton X100, 8 micromolar to the bilayer. And you don't need statistics to see that there is a large change in the monomer dimer equilibrium in the membrane. And when you looked at this, you sort of realized that, wait a minute, this is quite remarkable. And we did a number of studies where we tracked down what was the original reason for doing these experiments. But we looked at, among others, lipid metabolites, and we realized that a number of cellular signaling molecules had the same effect on the bilayer as Triton X100, and that is why I'm talking to you today. And so early on, when we did these experiments, we focused on amphiphiles much more than drugs. And Triton, for example, is a micelle-forming detergent. There are other molecules that you can add to a bilayer that in their own right would tend to form bilayers. And then there are molecules that would tend to form inverse micelles. These are molecules that have relatively small polar head groups, large voluminous hydrophobic tails like polyunsaturated fatty acids. And what we see here is that we see now structures with a positive curvature. Here we have no curvature. Here we have structures with a negative curvature. And if we take some of these molecules and then add them to a planar bilayer, if we add micelle-forming molecules, we would now produce positive changes in curvature. Here we would produce no change in curvature. And in this case here, with inverse micelle-forming molecules, we would have negative changes in curvature. And when we did these experiments in around 1988 and 89, the dominant picture of what was important for bilayer regulation of membrane protein function was curvature. And so the idea here is you take a bilayer and now you add molecules that either by themselves would produce positive changes in curvature or negative changes in curvature. And we now look at how will that affect the monomer-dimer equilibrium. And there's sort of a three terms that now becomes operational. I already introduced to you the disjoining force. There's a force with which the bilayer tries to pull the dimer apart. And if you have an increase in the disjoining force, then we will see that in the channel appearance rate, what is the frequency at which channels appear, and also in the lifetime. We would then expect that molecules that produce positive changes in curvature would reduce the disjoining force because it's easier to locally bend down the bilayer, and that'll lead to an increase in the appearance rate, an increase in the lifetime, and therefore cause a negative change in the free energy of the monomer-dimer equilibrium. And conversely, if we now add the molecule that produce negative changes in curvature, then we would expect to see an increase in the disjoining force, a reduction in the appearance rate, a reduction in the lifetime, and an increase in the energetic cost of forming the dimer. Experimentally, what we see here is we took two molecules, Triton X100 and capsaicin, and because of the results I'll show you in the moment, we felt it was critical to determine what were their effects on curvature, and which we did by studying the hexagonal repeat distance in small angle X-ray scattering experiments. And what we find is that Triton increases the hexagonal repeat distance, meaning they reduce they increase the diameter of these tubes, they cause positive changes in curvature, whereas capsaicin, which is the principle in hot pepper, it activates, as many of you know, TRPV channels. The valenoid receptor causes reductions in the diameter of these tubes, causes negative changes in curvature. And I should add, whereas I think there's good understanding of why Triton causes positive changes in curvature, nobody understands why capsaicin causes negative changes in curvature. We see that the changes in curvature vary as function of the mass fraction of the molecules in the bilayer. So when we now at that point, we did electrophysiological experiment, and we looked at what was the effect of Triton and of capsaicin, we would expect opposite effects. We have seen that Triton increases the likelihood of forming dimers, shifts the monomer-dimer equilibrium towards the conducting dimers. We would have expected, if curvature was a dominant feature here, that capsaicin should have the opposite effect. It does not. You see, again, capsaicin also shifts the monomer-dimer equilibrium toward the conducting dimers. And so we can therefore conclude that this popular scheme is wrong. Are the molecules that reversible amphiphiles that partition into the bilayer do not, in fact, increase the disjoining force. They reduce the disjoining force, and that is due to sort of rather deep thermodynamic reasons, that if molecules can freely equilibrate between the aqueous solution and the lipid bilayer, we increase the degrees of freedom that are available to the bilayer as it packs around a Gramocide channel, thereby reducing the free energy of the reducing the bilayer contributions to the free energy for the conformational change. Now, this is interesting as a purely biophysical point of view, because it raises a lot of questions. Some of them still remain unanswered, but you sort of have to ask yourself, is this just something that is a biophysical curiosity with the Gramocide channel, or is this a more general feature of amphiphiles. And so we would like to see what happens when we have integral membrane proteins. And we did experiments with voltage-dependent sodium channels that had been heterologously expressed. And here's the experiments at the top with Triton and at the bottom with capsaicin. And the way we did these experiments was that we tested for sodium channel function following an depolarization to 0 millivolts, but from a potential of either minus 130 millivolts, where the inactivation, which is an important part of a normal sodium channel function, has been eliminated, or from a potential of minus 60 millivolt, a potential at which the sodium channels are pretty strongly inactivated. We have, you see, the current, the maximal current, where after stimulation from minus 60 millivolt is less than half of that if we stimulate from minus 130 millivolts. And what we see is that if we add either Triton or capsaicin, that if we test for channel function from minus 130 millivolt, we have relatively modest inhibition of the currents. But if we test from minus 60 millivolts, we see a twofold inhibition of the currents, whether we do Triton or capsaicin. So there are two things that are important here. One of them is that it's not only with with Gramocide channels, but also with sodium channels that Triton and capsaicin have the same effects. So curvature cannot be important. And second, that the effect of both of these amphiphiles depend on the channel's functional state. That is, that both amphiphiles really alter the equilibrium distribution among different channel states. And this is a study from Rada Rusinova's work, where she looked, again, some of these detergents had been studied earlier by Jens Lundbeck, but she studied a number of drugs that have been used to treat type 2 diabetes. And what we find is that if we take and plot the changes in the midpoint for the inactivation curve versus the aqueous concentration, we will actually span 3 to 4 orders of magnitude in aqueous concentrations. But if we now, instead of using the aqueous concentration as sort of as a measure of activity, but the changes in Gramocide and channel lifetime, then we find that the results with all of these different molecules can be superimposed, at least for not too large changes in the Gramocide and channel lifetime. That is, that there seems to be a general change in the lipid bilayer properties that affect the gating not only of Gramocide channels, but also of the much more complicated voltage-dependent sodium channels that are embedded in a cellular membrane. And so if we now take a large number of different molecules, detergents, various drugs and other bioactive molecules, and either cholesterol depletion or enrichment, what we find is that there is a one-to-one mapping of changes in Gramocide channel function and changes in sodium channel function. So there is a generality to this. When capsaicin or capsaicin or adenosine does something to Gramocide channels, we actually can predict what happens to the changes in sodium channel function. By this, I do not mean to exclude the fact that there can be more chemically specific interactions, but the changes in the bilayer properties that we sense with Gramocide channels turn out to be an important regulator of sodium channel function. If we take a more limited number of molecules, then you see here that it's not just voltage-dependent sodium channels. We can have calcium channels, calcium-activated potassium channels, or various cis loop receptors. In some cases, there's no experimental results available in the literature, but again, you can see that we can use Gramocide channels to predict what is happening in the much more complicated ion channels reconstituted or embedded in cellular membranes. So And the basis for this, of course, is that membrane proteins undergo conformation, and that is a general feature not only of glit pH. Here you see structures of the KCSA, the first valence selective channel, whose structure was elucidated in Rod McKinnon's laboratory. The MESGES channel, the one I showed you earlier, a maltose transporter, and the calcium ATPase from sarcoplasmic reticulum. And you can there now make the general statement that at some concentration, almost any amphiphile becomes a modifier of bilayer properties, and meaning that it becomes an indiscriminate modifier of membrane protein function. But all proteins will not be equally sensitive to changes in bilayer properties, because the bilayer contribution will be the difference between the bilayer deformation energies associated with the different conformations. And so that sort of brings me now to the second half of the lecture or webinar, where we sort of now should begin to think about how can we actually pharmacologically alter an ion channel function. And there are 5 ways of doing it. We can occlude the pore, which in an enzyme would be occluding the active site. There will be allosteric control sites, as for example in the ligand-gated ion channels. There will be sites at the interface or where different subunits or even alpha helices get together. I'll show that in a moment. And then there will be other possibilities for pharmacological modification because molecules may accumulate at the interface between the bilayer and the protein, and thereby altering the detailed packing of the proteins around the channel. And finally, we will have sort of indiscriminate changes in bulk bilayer properties. If we now take the energetic formulation, we know that what we really care about at the end of the day is the total free energy for the conformational change. And we have that this allosteric control, of course, is exclusively reflected in delta G protein. These contribution will be reflected in the bilayer contribution, and the site 3 will be reflected both in the protein and in the bilayer. And here I show as a recent structure from Ryan Hipp's laboratory, which shows where general anesthetics are binding in the GABA channel. And you can see here that the general anesthetics indeed bind in regions that are similar to what we see here. And that will be now the contribution from the residual exposure, which is why I said earlier that these are likely to be pharmacological hotspots. But electrophysiology can get you a lot of information, but it's very slow. And Helge Ingelsson, who was a student in my laboratory a number of years ago, said that he actually would like to be able to study many more molecules and their effects on bilayer properties. And we developed a stop-flow fluorescence quench method. And here you see the hero in the story, the SX20, which we have put to good use for many years now. And the idea of the experiment is that we encapsulate a water-soluble fluorescent dye into the lumen of a large unilamellar vesicle. We have gramocide channels in the wall, or gramocide monomers and dimers in the wall of the channel, and gramocide channel happens to be very permeable to thallium ions, and thallium ions are heavy ion quenchers of the ANTS fluorescence, and therefore we can monitor the number of conducting gramocide channels in a LUV by the rate at which we quench the fluorescence here. And it turns out this process is fast, so we need the stop-flow capabilities in order to do the experiment. We have the fluorophore-loaded vesicles here that we mix with the thallium quencher, and then in the optical cell we follow the time course of quenching. And you see here we have our vesicle, and this shows a 1 second trace in the absence of quencher. with no gramicidin but exposed to quencher, and the gap here is due to the fact that we can get rid of only about 99.99% of the ANTS in the extracellular solution that is unavoidable when we load the vesicles with fluorophore. And then we see what happens when we work with increasing concentrations of gramicidin. We see increased rate of quenching, and we get more complete quenching because more vesicles have conducting channels. If we now take one fixed gramicidin concentration and now check what happens when we add a drug, and in this case the drug was capsaicin, we see that the increased concentration of the drug increases the quench rate, and again, because we shift the gramicidin monomer dimer equilibrium toward the conducting dimers, we will have more complete quenching of the vesicles. And so when Helgi took these experiments, then he, instead of doing one or two molecules, he actually could study a large number of linear and branched alcohols. And what he found was that if you go from methanol to hexanol, the bilayer modifying potency referred to the aqueous solution of the molecule, where it was inverse with the partition coefficient of the alcohols into the bilayer as estimated using C log P. What this tells us is whether it is methanol or whether it is hexanol, that the bilayer modifying potency per molecule of the alcohol in the bilayer is basically the same. If we increase the chain lengths beyond 6, however, then we see that the bilayer modifying potency per molecule becomes less, and that is now due to a competition between two different effects. The thermodynamic softening that we have discussed earlier, and then the fact that long acyl chains, like for example in decanol, will begin to reduce the mobility and the freedom of the acyl chains in the phospholipid bilayer, and thereby actually impose the stiffening try to, in its own right, to shift the monomer dimer equilibrium towards the non-conducting monomers. And we have done this with many different molecules, and here you see a list of the mains we have tried. We have done alcohols, various lipid metabolites, various neurotoxins, lipid metabolites, again here phytochemicals, and those in red, they are drugs that are used in human medicine. And overall, you can say that biologically active molecules tend to be bilayer modifiers. But what is surprising, what was very surprising to us in the beginning, was that if we now take these molecules and see what is a bilayer modifying potency for these molecules, all tested at 10 micromolar concentration, we see that molecules on average tend to be more bilayer modifying. As they become more hydrophobic, they are more likely to partition into the bilayer solution interface. But you see that the width of this sort of fan increases as we go out, and so that though the average, there will be more bilayer modifying, we can't really use log P itself to predict to what extent a molecule is going to be have perturbed the bilayer properties and therefore problematic in terms of its biological effect. And that sort of raises the more general question: bilayer modifying effects, are they friend or are they foe? And one thing is to study voltage-dependent sodium channels in isolation. Another is to study complex circuits, whether it is in a cell or sort of a neuronal circuit here. There are many players that are involved, and one therefore can sort of ask oneself the question: if you want to change the function of some more complex signaling circuit, do you really want to have 100% of the effects on one target, or would it be biologically advantageous and in a sense induce more stability if you distribute the effect over many different targets? We know that in cancer chemotherapy, there's a lot of discussion about polypharmacology, and that has served patients well. But the ultimate polypharmacology is actually if you go in and say you alter the properties of the bilayer, because that is going to alter the function of all of these proteins sitting here. It is non-specific in the sense that you might not want to change the function of this protein too much. You really want to focus on this one here. But you can still, modest bilayer modifying effects might actually be able to alter the function of circuits, say, involved in cardiac rhythmicity or in the central nervous system. And Rada Rusinova studied the effect of amiodarone, one of the most commonly used antiarrhythmic substances, which has no well-defined target. Indeed, it is known to alter a lot of different membrane proteins in cardiac cells that have all sorts of effects. And all of these are important in terms of amiodarone's ability to prevent serious cardiac arrhythmias, but we don't know actually which is the dominant one. And in addition to these effects that clearly affect the heart, then it has general antioxidant as well as anti-inflammatory processes. So this affects numerous different systems in our bodies. And Rada could then show, taking common antiarrhythmics, that they are all bilayer modifying, and at concentrations that are, when we now calculate what are the free concentrations that are similar to those that are used in clinical practice in the case of amiodarone and mostly amiodarone. And when we now see what is the relationship between the changes in quench rate determined using stop-flow fluorescence quench, we see that there are sort of characteristic fingerprints for each of these different molecules, and that allows us to conclude that some molecules, particularly amiodarone, and maybe dronedarone, really may involve a bilayer-mediated effect in terms of their desired clinical activity, whereas propranolol and pindolol are likely to act by mechanisms that do not involve the bilayer. Another example that I want to show, and then I will be done, is the long-standing question of how do general anesthetics work. Again, anesthesia involves changes in circuit function, but we have since the Meyer-Overton studies from the late part of the 19th century, early part of the 20th century. There's been this observation that anesthetic potency for many molecules correlated with their partition coefficient into various proxies for biological membranes. And so then this hypothesis that it was a lipid bilayer-based mechanism underlying anesthesia fell into disrepute, but there was never really a clear experiment that ruled out that showed that anesthetics could not alter function via the bilayer-mediated regulation that I've discussed with you today. And so, together with Hugh Hemmings, who is our chair of anesthesiology, Carl Herold, a postdoc in his laboratory, and Leah Sanford, a student in my laboratory, did extensive studies on clinically used anesthetics and a few other molecules, and showed that yes, all of these molecules, with a couple of exceptions, are bilayer modifying. But if you look at the bilayer modifying potency, you see that they're only bilayer modifying at concentration so high that they will be toxic. And if we now take compare a lot of different molecules on different ion channels, then we have, and we use the changes in quench rate as a measure of their bilayer modifying effect, then we see that there are sort of honest to goodness bilayer modifiers, as you see here, where we have capsaicin and other molecules, and they can cause changes in membrane protein function that indeed are related to the changes in quench rate, but that anesthetics, the clinically used anesthetics, basically insert their effects with minimal changes in Gramocide and channel function, and we therefore can conclude and sort of probably the final nail in the coffin whether there is a bilayer-mediated effect in anesthesia, that anesthetics, they by exclusion work by binding to specific protein targets, not by modifying in a non-specific manner lipid bilayer properties. So that brings me to my take-home messages, that bilayers regulate membrane protein function because membrane proteins undergo conformational changes. I think something that I think is actually quite remarkable, it is possible to use single component bilayers to study drug-induced changes in membrane protein function in cellular membranes, as I showed you with the studies on voltage-dependent sodium channels. That is, we have one phospholipid. Cell membranes may have 400 phospholipids. But at the end of the day, a lipid bilayer is a lipid bilayer. Reversibly absorbing amphiphiles alter lipid bilayer properties. When the changes in bilayer properties become sufficient, these molecules cause indiscriminate changes in protein function. And this may and most likely lead to undesired toxicity, but in the case of amiodarone, for example, very likely desired polypharmacology. And all amphiphiles will alter bilayer properties at some concentration. And so you really need to be careful what are the concentrations that you use when you study the biological phenomenon of hand, and what are the concentrations where the molecules you use alter bilayer properties. The bilayer modifying potency is in general not predicted by hydrophobicity, although hydrophobicity does provide guidance. And also, there's sometimes people are worried about using small hydrophobic molecules to manipulate biological function. It's perfectly okay to do so, but you need to know what you're doing. That is, you need to know what effect do these molecules actually have on lipid bilayers at the concentrations where you use them. And I thank you for your attention.
Thanks for the thanks for that, Olaf. That was very interesting. I'll ask a question. The stop-flow assay that you carry out to get this data, what are the biggest challenges associated with that assay?
I would say that for the stop-flow assay, it is a much simpler assay technically than the electrophysiological assay. It takes time. The electrophysiology takes time to learn. The stop flow, I would say to some extent sinks to the quality of, and this is not something I'm being paid to say, by the way, but sinks to the quality of the SX20. It's a wonderful machine. It works, and you need to maintain it, and you need to not only sort of on a daily basis, you need to keep it clean and so on. But once you have it, it is an amazing system. The largest challenge is to form vesicles that are so tight that you know that you are only observing thallium, or you are almost exclusively observing thallium influx through the Gramocide channels. Because if there is movement of thallium ions across the lipid bilayer that is not catalyzed by Gramocide channels, it would lead to a background flux that becomes leads to more complicated analysis of the experimental results. But otherwise, it is a simple, robust, and incredibly reproducible and reliable assay.
Great. My other question was, where does it go from here? What are the next kind of steps for you?
I think the next steps are, and we have been, I didn't show any of this, is two. One, there is a certain amount or degree that it is still, you can say, empirical. I think we have a decent understanding of the theory of elastic bilayer deformations, but we can still improve that. I think it is remarkable to me, if you look at the alcohol experiments, that per molecule in the membrane, that a hexanol seems to be as good or bad as a methanol molecule. I mean, there's something that we still do not understand there. And that was actually the reason we started out with developing this fluorescence method, because the single channel electrophysiology or any electrophysiological method would not allow us the throughput that we would need in order to develop a database that, for example, would allow us to test computational models of small molecule modification of lipid bilayer properties. So, and one of the things we have done over the more recently, I didn't discuss today because it would sort of lead to a longer talk, is that we are taking these concepts and we are now moving them into studies where we examine cytotoxicity, that is, what the relationship between the toxicity of small molecules on cells in different contexts and the bilayer modifying potency. And we find that we are actually able to really predict with remarkable ability whether a molecule is likely to be cytotoxic. I can tell you that if a molecule changes the quench rate by more than a factor of 5, 100% of the molecules will be toxic. Molecules may have no effect on the bilayer and they will still be toxic because they can be toxic for any number of reasons. But bilayer modifying ability, bilayer modifying potency becomes a strong predictor of cytotoxicity, which I think is likely to guide future drug development, because this is a mechanism. I mean, despite what I just said a moment ago, this is a mechanism-based assay. We know that it reflects the ability to alter bilayer properties, reflects the ability of the molecules to partition into the bilayer. And therefore, if you, for example, take propranolol and pindolol, two of the beta blockers I showed, propranolol has an anthracene and pindolol has an indole, and indoles have much lower partition coefficient into lipid bilayer membranes than the anthracene, and therefore you would probably a priori expect that pindolol might be less bilayer modifying, but it is one of these things that you have to know in order to use it as a guide going forward. And so one of the things we are doing right now, and it will demand a larger, still a continued experimental effort, is to develop a understanding of how very diverse group of molecules will alter lipid bilayers, and based on that, use various deep learning neural network tools to see, can we actually establish rules for when molecules will be bilayer modifying. Despite what I said, I showed you this fan. sort of that become wider and wider when we looked at various drugs and their effect on bilayer properties. Hydrophobicity is not terrible in predicting bilayer modifying effect, but it's not sufficient. And I think we may be able to develop such a guide for when molecules are bilayer modifying. But of course, nobody would ever fully trust them. So you would always want to go back and do the experiment. The neural networks will provide important information going forward, but I'm not going to work myself out of a job anytime soon.
I have two more quick questions before we finish. The first one is from the audience. Have you ever characterized the effect of typical inverse surfactants such as AOT in the formation of Gramocide in channels?
No, we have not. The thing we have done, that's a good question, and it is an obvious one we just haven't done. But we have done polyunsaturated fatty acids, capsaicin, those are the major, and then also one molecule that continues to confuse me, olelamine, which is I would expect to behave like any charged amphiphile, but oleic acid, the deprotonated, the oleate, and olelamine have completely opposite effects on the bilayer for reasons I have no understanding of whatsoever. Rada did some really thought-provoking experiments there, because with long-chain molecules, we actually find that their effects on the bilayer depends strongly on their charge. Part of that we can rationalize by the fact that the charged molecules will have higher solubility in the aqueous solution. There will be a better defined equilibrium between the membrane and the bilayer, but other things we really do not. But thank you for the suggestion. AOT is something we should be looking at.
Awesome. The final question is, have you and your team worked with non-lipid based vesicles such as block copolymers for use in your assays? And if so, how do they affect membrane protein activity in your studies?
We have not done that at all. I've been thinking about it, but I would say up to now, and probably it will continue, lack of time is a significant factor, because my interest is really, so to say, trying to get as much insight as we can into sort of the regulation of membrane proteins in cell membranes. And I agree that the block copolymers would be a very interesting thing to pursue, but I'm not sure that they will percolate to the top of the priority list anytime soon, because, well, just lack of time.
Fair enough. Well, all that's left for me to say is to thank you again for your presentation, Olaf, and taking the time out to speak to us today. I'd like to thank everyone who's attended for also taking the time to come along and join us. Hope you got something good out of the webinar. If you do have any questions that we didn't get around to answering today, or you didn't get around to asking, then you can email those into us at info@photophysics.com, and we'll gladly put those across to Olaf for you. All that's left for me to say is if you'd like to come along to the next webinar on the 8th of April at 11 AM Eastern or 4 PM British Summer Time, and then please go look at the website photophysics.com to get more information and register.
Yeah, and so Tom, thank you very much for the invitation to speak. And I sort of second your comment that I hope the audience got something out of it.
Yeah. No, thank you very much for coming along and helping us out today. All that's left for me to say is thanks for everyone for coming, and enjoy the rest of your day.
Thank you.